Hello everyone, welcome back to our channel, and today we'll be learning about one of the oldest and most versatile molecular biology technique, the polymerase chain reaction or PCR. PCR was initially proposed by Dr. Hargobind Khurana in the 1970s. However, at that time it was more of an art than science. 15 years later, Cary B. Mullis independently conceived and executed the polymerase chain reaction process, for which he won the Nobel Prize in Chemistry in the year 1993. So, what is PCR? As the name suggests, polymerase chain reaction is a chain like repetitive or continuous process which helps in the generation of multiple copies of DNA from a single target sequence. Essentially, this process could be broken down into three distinct steps: denaturation, annealing, and extension. And these three steps could be repeated for n number of times or for n number of cycles, depending upon the final concentration of products which is desired. So let's break this down. We know that DNA is double-stranded with the bases held together by hydrogen bonding interaction. So adenine and thymine are bound together by 2 hydrogen bonding, whereas guanine and cytosine are held together by 3 hydrogen bonds. In the step of denaturation, high temperature is applied to the double-stranded DNA, which causes the breakage of these hydrogen bonds and dissociation of the double-stranded DNA into two separate strands. So generally, the temperature of denaturation ranges from 94 degrees Celsius to 98 degrees Celsius, depending on the polymerase which is used for the reaction. Now, after dissociation of the double-stranded DNA into two separate strands, a pair of oligonucleotide primers are allowed to anneal or hybridize to these single-stranded DNA. Again, this hybridization is driven by Watson-Crick base pairing. And finally, extension, where the polymerase keeps on adding nucleotide to the newly synthesized strand based on the template DNA sequence. So the extension generally takes place at a temperature of 68 degrees Celsius to 72 degrees Celsius, again depending on the polymerase which is used for the reaction. So let's have a look into the sequence of amplification in the PCR. So initially we begin with a single copy of a double-stranded DNA. And these two regions marked in yellow shows the desired region we want to amplify from this double-stranded DNA sequence. So this DNA is subject to denaturation at a high temperature, followed by annealing of the primer. So this is my forward primer and this is my reverse primer. So let me label this for you. So here is my forward primer and here is my reverse primer because it is moving in the reverse direction. Now, as you can see in the extension step, the polymerase is incorporating nucleotide, leading to the extension of this primer depending upon the sequence of my template DNA strand. Always remember that the polymerase moves from the 5' end to the 3' end. So if I label the sequence for you, this was my original double-stranded DNA. And this is the sequence of my newly synthesized DNA. So far, so good. So this is my first round of PCR amplification. Now what happens in the second round of PCR? Again, at high temperature, all the four strands dissociate, followed by the binding of the forward primer and the reverse primer, followed by extension of these primer sequence, leading to the generation of 4 pairs of DNA strands. This cycle is again repeated for the third time, where all these 8 strands separate, followed by the annealing of the primer and the extension. Now, if you look carefully, you would realize that all these strands are heterogeneous because this primer extends beyond the site which we want to amplify. Similarly, the forward primer also extends beyond the site we want to amplify. So this is a heterogeneous population of double-stranded DNA. Similarly, in the third round of PCR, we get several heterogeneous population. Apart from these two DNA strands, where we are actually getting our desired amplicon or PCR product, where the end of the DNA sequence is basically determined by the 5' end of the forward primer and the reverse primer. So always remember that when amplifying a small segment of double-stranded DNA, the desired PCR product or the target sequence begins to increase exponentially or accumulate exponentially from the third cycle of PCR. So basically, we get a homogeneous population of our target population from the third round of PCR. So in cycle number 1 and cycle number 2, there is no amplification of the double-stranded target molecule. There is a heterogeneous population. From the third cycle, the exponential accumulation of the target DNA sequence begins. Now, this reaction is mediated by the polymerase. An important characteristic of this polymerase, which catalyzes the polymerization reaction, is that it needs to be heat stable. Because as I have mentioned, the denaturation, annealing, and extension takes place at high temperatures, like about 70 to 90 degrees Celsius. Now, most of the enzymes with which we routinely work in our laboratories are active at a temperature of about 25 to 40 degrees Celsius. So if this is the scenario, then for every cycle, we need to freshly add enzymes into our tubes. But we do not need to do this in a thermocycler machine. This is because of the discovery of a thermostable DNA polymerase, which remains active at high temperatures of about 90 to 95 degrees Celsius. And the first thermostable DNA polymerase, which was isolated and most commonly utilized in all laboratories, is the Taq DNA polymerase, which has been identified from Thermus aquaticus. So Thermus aquaticus is a thermophilic bacteria which has an optimal growth temperature of about 70 degrees Celsius. So as you can realize, all the enzymes of this organism is tuned to work at extremely high temperature. And this bacteria was isolated from a hot thermal spring at the Yellowstone National Park. And the discovery was done by Thomas D. Brock, who was a microbial ecologist. So what are the essential components of a polymerase chain reaction? Most important is the thermostable DNA polymerase. Now, as I have said, Taq polymerase is the most commonly used polymerase in most laboratories for routine amplification process. While choosing a polymerase, two factors need to be considered. One is the polymerization rate, that is the number of nucleotide the polymerase is adding to the elongating primer per unit time. So normally for Taq DNA polymerase, the polymerization rate is around 1 kb per minute. So in 60 seconds, it adds 1000 base pairs to the elongating DNA. And the other factor is the fidelity of the enzyme. That is the accuracy with which it is adding the nucleotide. Now, as we would see in subsequent chapters, there are several other polymerases identified which has better proofreading activity than Taq polymerase. And those are generally used when you are trying to amplify DNA sequences of about 5 kb and larger, and you do not want mutations to be incorporated in your amplified molecule. So two factors need to be considered: the polymerization rate of the enzyme and the proofreading or the fidelity of the enzyme. Next, buffer. We know all enzymes need a buffer to function optimally. So most polymerases come with their own proprietary buffer mix, which is usually Csir. Now, in the vial, the pH of the buffer is generally kept at around 8.3 to 8.5. You know that with an increase or decrease in temperature, there is a fall in pH of a buffer. So at room temperature, the pH of the buffer is around 8.3 to 8.5. However, when the PCR reaction takes place at high temperature, the pH of the buffer falls to around 7.2, which is the optimal pH for the activity of the DNA polymerase. Also, most enzymes require cofactors for their activity. DNA polymerase or Taq polymerase is known to function in the presence of divalent cation like magnesium, which is normally supplemented with the buffer at a concentration of about 1.5 millimolar. But you can fine-tune your experiment by altering or adjusting the amount of magnesium. Third, the template DNA. Definitely you need a template sequence to which the primers would bind and extend. So it has been seen that linear DNA is amplified more efficiently than circular DNA. And if you have a high molecular weight DNA, if you chop it down by digestion with restriction enzyme, the efficiency of PCR amplification has been seen to be greater. What is the amount? PCR is a highly sensitive process. So even picogram to low nanogram levels of template is sufficient for the amplification. So theoretically, even if you have a single copy of your target sequence, PCR reaction would allow the exponential accumulation of that product. But normally in our routine laboratory setup, we use around picogram to nanogram level of the template DNA. Another crucial factor which determines the accuracy of a PCR reaction is the pair of synthetic oligonucleotide primers. Now, these primers should be designed in such a way that it not only amplifies the desired product, that is the specificity of the reaction, but also it should suppress unwanted amplification of DNA. So there are several factors which need to be kept in mind while designing primers. First is the base composition. Normally, a higher GC content of about 40 to 60% is preferred because, as I have said in my previous slide, guanine and cytosine are held together by 3 hydrogen bonds. So greater the GC content of a primer and the more uniformly they are distributed across the primer sequence, greater would be the binding efficiency of the primer to the template DNA. Also, increasing the GC content increases the melting temperature of the primer. A higher melting temperature ensures that there is no non-specific binding of the primer to the template. Third is the length of the primer. Usually, primers are kept at a length of 18 to 25 base pair. If you design a very short primer, there is a chance that it would bind non-specifically or randomly to anywhere in the large genomic DNA. On the other hand, if the primer is too long, again, the binding efficiency of the primer gets compromised. So it has been seen experimentally that primers of length 18 to 25 base pair are best suited for PCR amplification. Fourth and fifth is there should be no formation of homodimer or heterodimers in the primer. What do I mean by homodimer? If there is self-complementarity or repeated sequence within the primer sequence itself, it will have a tendency to form a hairpin-like structure. Now, these hairpin-like structures are very stable and they would basically sequester the primer and not allow it to bind to the template DNA. So while designing the primer, you have to be very sure that there is no complementarity in the primer sequence itself. And the second mode of complementarity is between the two primers, that is between the forward primer and the reverse primer. If the forward primer and reverse primer anneals to themselves, it would form a heterodimer duplex, which is again extremely stable and again prevent the primer from binding to the template DNA. So normally there are several computational tools which helps in determining the efficacy of a primer. An example of such a tool is oligoanalyzer, which is maintained by IDT. So you can have a look into the website. And finally, DNTPs. So the polymerase requires the nucleotides which it would add to the elongating strand in a template dependent fashion. So an equimolar concentration of the four nucleotides DATP, DTTP, DGTP and DCTP is used in a PCR reaction. Again, you should remember that there is no hard and fast rule for PCR. The concentrations which I have mentioned over here are seen to be applicable for almost all routine amplifications which we do in our laboratories. However, depending upon your need, you can manipulate with the concentration and amount of the components of a PCR reaction. So there are several applications of PCR, of which I am mentioning the major three. One is the molecular biology research. For if you're interested in identifying a gene sequence, you want to clone it and study the expression pattern of a particular gene, PCR would be your first step. So it is an indispensable tool for molecular biology research. Second is medical diagnostic, where it is used for the identification of infection pathogen, be bacteria or virus. If you remember that during the COVID period, we had a PCR-based COVID test. So what was that? Basically, you take the sample from the patient. The RNA is isolated from the patient sample, be it the throat swab or the nose swab. So instead of DNA, in this case, RNA serves as the template, which we would be learning during RT-PCR lessons. So the RNA is isolated from that sample and it is converted to cDNA. And you add specific primers which could recognize the viral specific molecules. So if the primers have binding sites in the DNA of the isolated from the infected patient, you would get a band and the patient is said to be COVID positive. However, if the patient is not infected, the primers will not find any binding site in the isolated RNA and you will get a negative result. So it is an indispensable tool for medical diagnostics as well. And finally, forensic science. So, for example, in a crime scene, you get a very minute sample of the suspect, a hair sample or a nail sample. So what you do is you isolate the DNA from that, you use specific primers and you compare the profile or the DNA profile, it is also called DNA profiling, of the hair or the nail sample to the DNA profile of the suspects. So there are three suspects, suspect 1, 2, and 3, and to compare the DNA profile with that minute sample you have discovered from the crime site. The one with which it matches is your usual suspect. So these are some of the major applications of PCR. Apart from this, it is also used in archaeology and paleontology studies, as you have seen in the Jurassic Park movies. So in the subsequent lessons, we would learn about what are the different kinds of PCR, and most importantly, what is RT-PCR, which helps in the quantitation of the amount of DNA amplified. Thank you.